Hi guys, today in this video we will complete talking about section 4.6, which is fermentation. Firstly, what's the meaning of fermentation? Fermentation is an anaerobic process which means doesn't require oxygen. This process allows glycolysis process to be continue. In case of absence of oxygen, we know that glycolysis process means a splitting of glucose, one molecule of glucose, into two molecules of pyruvate. So we can say that glycolysis process, in case of a present of oxygen, which means with oxygen, so it's complete the cellular respiration process for the production of energy. But in case of absence of oxygen, without oxygen, so this process will complete as fermentation process. Let's talk about some examples about fermentation process. We have two main examples in your curriculum: lactic acid fermentation and alcoholic fermentation. Let's talk about some notes of this process. The first point, you must know that in this process fermentation doesn't make ATP, but allow glycolysis to be continued. But this doesn't mean that fermentation process is not share in the production of energy, but this process doesn't make ATP directly. The second point is fermentation process removes the electrons from the carrier of electrons like NADH, and we know that when the carrier of electrons NADH loses its electron, it becomes NAD, which carrier of electrons. So why fermentation process removes these electrons from NADH, and why this process is important? So pay attention please. Glycolysis process, we know that it's similar to cellular respiration, which needs electrons to each molecules have the ability to pick up electrons. This molecules like NAD, NADP, FAD, but you must know that NAD, and FAD, they are carrier of electrons during cellular respiration, but NADP carrier of electrons during photosynthesis process. So fermentation process removes the electrons of NADH to P NAD, which help glycolysis process to complete and to be continue in case of absence of oxygen. Let's talk about the lactic acid fermentation, the first example of fermentation. Suppose that molecule of glucose has been split exactly by glycolysis process in one of your muscles, but during this glycolysis or splitting of glucose, oxygen is unavailable. So what will happen? Excellent. This process of glycolysis will be stopped. Why? Because there is no oxygen. So what will happen? The muscle cells required and needs energy. So in this case of absence of oxygen, the glycolysis process must be continued. So process of lactic acid fermentation takes place. Okay, so lactic acid fermentation takes place inside vertebrates muscles, inside some microorganisms cells. Vertebrates cells, muscles are cells, but you must know that in general, not just muscles, all cells of vertebrates and some microorganisms cells. The chemical formula of lactic acid have 3 carbon atoms, so its chemical formula C3H6O3. What about the steps of lactic acid formation? Lactic acid fermentation. Firstly, we said that glycolysis occurs, and to be complete or continual fermentation process takes place. So glycolysis means splitting of glucose, and we know that as a result of splitting of one molecule of glucose, 2 molecules of pyruvate produce and 2 molecules of carrier of electrons NADH produce. So the process of fermentation, lactic acid fermentation, used these materials, which is pyruvate and NADH, from glycolysis to enter its steps. The 2 molecules of NADH, provide energy. This energy used to convert pyruvate into lactic acid. Again, the 2 NADH, which produced from glycolysis, used to provide energy to convert pyruvate into lactic acid. As a result of using this 2 NADH, they will be just 2 NAD without electrons. So this molecule NAD recycled back to glycolysis process, which allow the glycolysis process to continue. Again, lactic acid fermentation used the pyruvate and NADH, which produced from glycolysis. NADH provide energy, which convert pyruvate into lactic acid. So as a result of this convert of pyruvate to lactic acid by using NADH, NADH becomes NAD, and this NAD molecule are recycled back to glycolysis process, which allow glycolysis process to continue. If you want to make a simple summary for glycolysis and fermentation, we can show it like this. We know that the glycolysis start by splitting glucose, and we know that glucose have 6 carbon atoms, and we know that we used 2 molecules of ADP, 2p production of 2 ATP, and as a result of splitting of glucose, the electrons must be carried by NAD, and when NAD carry electron, it becomes NADH. And pay attention, please. So as a result of glycolysis of 1 molecule of glucose, 2 NADH produced and 2 pyruvate produced. So this process called glycolysis. Then the fermentation process occurs, but you must know that this process occurs in case of absence of oxygen. So now oxygen is unavailable. So what will happen? The pyruvate and NADH, which are produced from glycolysis process, used in fermentation process. How this 2 NAD convert pyruvate into lactic acid, which have 3 carbon atoms, and 2 NADH converts into 2 NAD. This NAD, this 2 NAD recycled back to glycolysis process to continue. Okay guys, thank you.